Okay, folks, today we will be learning about the electron transport chain. And so today and next time we're together, we're going to finally wrap up oxidative metabolism and have a better understanding now of why redox molecules are considered high energy compounds. We will walk ourselves through all the steps of the electron transport chain. We will have an understanding of where electrons from glycolysis come into the electron transport chain. Remember, those electrons are coming from the NADH that glycolysis produces. We'll understand why movement of electrons through the electron transport chain releases energy and how that energy that is released is conserved. We'll understand proton pumping that happens in complexes 1 and 4. We'll look at many different types of electron transfer and many different types of cofactors that can do electron transfer. We will understand what the purpose of the Q cycle is in complex 3. And we will eventually, folks, remember the big picture here is to link these pathways together and think about flux. So eventually we'll be able to think about flux through the electron transport chain and how it might be changed based on what is happening in the rest of the electron transport chain. And then pretty soon, what happens to flux through the electron transport chain if we mess up glycolysis, the TCA, for example. Keep in mind that there is an electron transport chain handout that might be helpful to you. Today's Meet a Scientist moment is Ada Almuteri. She's an associate professor at the University of California, San Diego. She does materials engineering in the realm of metabolism. So how can we engineer materials to visualize tumors or to study inflammation? Or how do we improve MRI technology, for example? Let's start with an introduction of the electron transport chain, or ETC. So we have moved our way to the inner mitochondrial membrane, folks. So the TCA happened in the mitochondrial matrix. Now this is happening kind of at or in the inner mitochondrial membrane. Some of the proteins we'll discuss are integral membrane proteins, and so they're in that membrane, and then a couple of the things we'll talk about exist in the inner membrane space of mitochondria. Let's keep in mind, folks, that movement of electrons from low to high reduction potential, releases energy. So just know that the sequence of proteins and cofactors in the electron transport chain is roughly ordered by reduction potential. Overall, folks, the process of moving electrons through the electron transport chain, starting either at NADH or FADH2 and moving eventually to oxygen to form water, releases a lot of energy, about 218 kilojoules per mole. So we have a lot of energy being released here. But if all of this energy were released at a single point, folks, it would be a challenge then to synthesize ATP. Each ATP only requires about 30 kilojoules per mole. But if we're releasing all of this energy at once, most of it would be wasted because we'd only synthesize a single ATP. So what the electron transport chain does is it breaks down the transfer of electrons into these various complexes so that the amounts of energy released can be broken up and more ATP can be synthesized, right? So rather than all of this energy being released at once, we see that energy is released in complex 1, energy is released in complex 2, in complex 3, and complex 4. Enough energy is released in complexes 1, 3, and 4 that each of those energy release steps more or less corresponds to the production of an ATP. So the electron transport chain breaks up energy release in 2 steps to conserve more energy as ATP. So we break down this massive energy release into multiple steps so that at each of those points, we've released enough energy to synthesize an ATP. Over the next few classes, we may be talking about inhibition of various complexes, folks. So keep in mind here on the screen, I've showed you some various inhibitors of the complexes. Rotenone and amytal inhibit complex 1, antimycin A inhibits complex 3, and cyanide inhibits complex 4. So just know there are inhibitors of these complexes. And before we jump into learning in more detail about the electron transport chain, I will reference this picture. This picture will probably be helpful for us to use along the way because it is a summary of the electron transport chain. It shows how NADH comes in and gives its electrons, how FADH2 comes in and gives its electrons, and then the overall flow of electrons eventually to oxygen to form water, and then also the flow of protons that will be important for us in our discussion today. Up next, let's walk ourselves through each of the complexes.